I'm Anthony Baldor, Vice President, Corporate Strategy and Development at 4D Molecular Therapeutics. And at 4D, we are harnessing the power of directed evolution for targeted gene therapies. Okay, this presentation contains forward-looking statements. All right, let me start off with a few vital stats on the company. Dave Kern and Dave Schaefer co-founded 4D about 8 years ago. The company is based in Emeryville, California. We have both research and GMP manufacturing facilities on campus. The platform is based on 2 decades of Dave Schaefer's work in directed evolution of AAV capsids. And today we boast a library of over 1 billion capsid sequences across 40 libraries. The pipeline is diversified with multiple development programs across 3 therapeutic areas, and we now, including 2 INDs, which we announced last week, have 5 gene therapy programs at clinical stage. Q4 data. We released initial clinical data for our XLRP program yesterday, and we anticipate release of initial clinical data for the Fabry program later this year. And lastly, we do intend to build a fully integrated biopharmaceutical company, including commercial capabilities. So everyone in this room likely understands AAV, relatively safe, somewhat effective at delivering DNA, and it's these qualities that have enabled some successes, including approved biologics. However, AAV have major limitations, including limited delivery to target tissues, limited transduction of target cells, inflammation and associated toxicity, inhibition by pre-existing antibodies. The limitations have forced the field to narrowly focus on some niche opportunities, primarily in rare diseases. But we at 4D believe we can overcome these limitations and bring gene therapy to millions of patients. So over 20 years ago now, co-founder Dave Schaefer started building on Dr. Francis Arnold's now Nobel Prize-winning work by applying directed evolution, which Dr. Arnold calls the most powerful biological design process, to both viral and non-viral gene therapy vectors, including AAV. At 4D, we have now industrialized directed evolution of AAV through our platform, which we call therapeutic vector evolution. The next 2 slides cover the 3 major steps we take to invent targeted evolved vectors for gene therapies. First, we create massive diversity of AAV capsid proteins using a range of molecular biology techniques. I mentioned now we have 40 libraries with over 1 billion capsids. So second, we set a TVP, a target vector profile. The parameters include target dose, route of administration, target tissue, and in some cases, antibody resistance is important. And third, we run an iterative competitive selection process to identify the fittest vectors matching that TVP. We do all this in NHPs, non-human primates, our closest animal relatives, and we've completed 15 target vector profile selections to date, filed over 300 patents on those capsids. With the allowance of our CF-IND last week, all 3 vectors are now at clinical stage. So once we've invented a lead vector, we design and engineer product candidates, which are tested in both NHP and human cells before entering the clinic. Importantly, we advance multiple product candidates using the same lead vectors, often in parallel. AAV is a highly modular technology. Through this strategy, the company captures pretty significant savings of time and capital for development. Okay, leveraging the platform and its modularity, we've developed a diverse pipeline for both rare and large market diseases, including broad patient populations, which we believe our competition will have trouble or be unable to address. Our initial product candidates are focused in ophthalmology, cardiology, and lung. Each of our product candidates is built on a proprietary targeted and evolved vector, and we believe the diversity of our pipeline may contribute meaningfully to our potential success. So again, now 5 clinical programs, initial XLRP data released yesterday, and initial February data planned for later this year. Okay, we have limited time, but let's at least take a quick look at the ophthalmology portfolio. Several programs in development here, all based on lead ophthalmology vector R100. All right, well, all major biologics for the eye are administered by intravitreal injection. AAV has been generally limited to subretinal surgery, with a few exceptions. And with the subretinal approach, the vast majority of the retina is not treated. So on the next slide, I have an image showing how subretinal delivery is confined to a bleb around the injection site. But with this backdrop, we invented R100 for intravitreal delivery, efficient treatment of the entire surface of the retina, and transduction of all major cell layers within the retina. This profile may allow us to treat a wider range of retinal diseases, including rare and large patient populations in all stages of these diseases. So on the left, bottom left, this image captures the unique structure of our proprietary R100 vector. In the upper right, we illustrate the limitations of conventional AAV as delivered by intravitreal injection. Conventional vectors like AAV2 are trapped and blocked by barriers, including the eye's inner limiting membrane. In the lower right, we illustrate how R100 was designed for penetration of these barriers, as well as for improved transduction of retinal cells. And we've confirmed in NHP that R100 achieves what we intended for it to do. So on the left, these images show the differences between the subretinal and intravitreal injection techniques, along with marker transgene expression in life images of the retina in primates. In the upper left, we see typical subretinal bleb and retinal detachment after subretinal surgery. This is indicated by the dotted line. In this example, McLaren et al. Reported that less than 1% of the retina was treated through that route of administration. In marked contrast, in the lower panel, in light blue, we demonstrate that intravitreal R100 is able to transduce cells across the entire retinal surface. Now, these in-life camera images don't show the entire retina, so the image on the right illustrates the estimated difference in coverage of the retina with intravitreal R100 in light blue and the subretinal surgical bleb with conventional AAV in yellow at the center. I think this is a detail that's often sort of missed. Because subretinal AAV only treats a tiny fraction of the retina, these therapies are limited to patients with very advanced disease with only a small remaining portion of retina remaining in and around the fovea. Okay. Oh, in contrast, R100-based products treat the entire retina. So this will hopefully allow us to prevent vision loss in a wider range of patients, including those in the earlier stages of their disease. This slide, a little bit of data, shows clear proof of principle for transduction with R100 in both NHP retina cells and in human retina cells. On the left, we have GFP expression in red from a typical NHP following intravitreal R100 carrying the GFP marker gene. The image shows widespread transduction and gene expression through all major cell types and in all regions of the retina, from the fovea on your right to the retina periphery on the left. And the right panel, we demonstrate superior GFP transgene expression in human retinal epithelial cells with R100 as compared to conventional AAV across a range of drug concentrations, or MOI as it's referred to here. So we confirm our vector transduces human cells. With the remaining time, I want to touch briefly on two of our key clinical programs. 4D125 is our clinical stage product for XLRP. XLRP is a rare disease that causes progressive vision loss and blindness. There are no approved therapies. The estimated prevalence is 24,000 patients in the US and the EU, in the 5 major EU markets. 4D125 was designed to treat patients at all stages of their disease, including patients in the early stage. 125 is, of course, an intravitreal product and is therefore highly differentiated from the competition, which is all subretinal. Excuse me. Okay, that's better. The candidate is currently in a phase 1/2 dose escalation study where patients are followed for safety, tolerability primarily, but also biomarkers of activity. And these biomarkers include microperimetry for visual field function and anatomical measures of retinal viability. And there are now, as of yesterday, preliminary data available, which we presented at ASRS and in a conference call. And I wish I had those data here, but we submitted this presentation before the data were available publicly. They were released over the weekend. So quickly, the takeaways for that data set are 4125 was well tolerated with no dose-limiting toxicities, no serious adverse events, and no chronic inflammation. We saw evidence of clinical activity in the treated eye versus the untreated eye in all evaluable patients and across 3 different clinical endpoints, including anatomical measures and measures of visual acuity. We're continuing to enroll patients at the high dose and in an expansion cohort, and that will include patients with less advanced disease. So please go and find the release for those additional details. There's likely a recording of that presentation available as well. Okay. A second important development for the 4D ophthalmology portfolio in the last week, our IND for 4D-150 for the treatment of wet AMD and DME was allowed. Given the preliminary tolerability profile we're seeing with 125, the XLRP candidate, we're thrilled to be advancing this 4D-150 wet AMD DME candidate. Importantly, the 150 product will enter the clinic at a dose which is only 3% of the 125 dose. The current treatment for wet AMD and DME is frequent intravitreal injection with anti-VEGF proteins, and there's a high unmet need remaining because the frequency of injections has translated into patient adherence issue, which has resulted in a high number of incomplete responders. The incidence and prevalence for these diseases is very high, and 4D-150 is well differentiated as a single-dose intravitreal therapy that employs 3 mechanisms of action. First, aflibercept is expressed to inhibit VEGF and placental growth factor, and a second transgene inhibits a third angiogenic factor. All right, because wet AMD and DME are more common diseases with approved biologics available, the bar for safety is higher as compared to rare diseases such as XLRP. This slide is to highlight that on top of the favorable safety profile we have recorded to date for 125 XLRP program and the lower doses planned for the 150 program, we've now conducted 3 GLP tox studies with R100, including a total of 91 primate eyes. We report no adverse findings in any of these studies. The treatment was well tolerated. No cellular immune responses were detected to either the vector or the transgene product in these studies, and the data gives us confidence in the tolerability of R100 as we initiate our first clinical program with a large market product candidate. Now, VEGF is certainly a validated target in these indications, and our base molecule is included in an approved product, but we ran a preclinical efficacy study with 150 in the choroidal neovascularization model of wet AMD and NHPs. We treated animals 7 per group with intravitreal 150 at 3 doses. Animals received steroid treatment for 28 days following administration, remained off steroids for the remainder of the study, and any uveitis resolved prior to the end of the 4 weeks of steroid coverage. No adverse findings were reported in life through 6 months of follow-up. And the chart here shows all animals experienced complete suppression of neovascularization, all 3 dose levels. So that's a little proof of principle for the 4D-150 product. And now, keeping it under 15 minutes, I'll just conclude by thanking our tireless team at 4D. It's been and should continue to be an incredible year with 5 programs now in the clinic, 2 initial data readouts, 1 last weekend and 1 more to be shared later this year, and 2 new INDs allowed in the same week last week. So thank you very much for your attention, and you can come and find me, because I'm running out of time, at the back of the room if there are questions. Thanks very much.